4. Okay, kind of act a little bit more enthusiastic. Okay. Okay, it is my task this afternoon to introduce to you Dr. Jayne Morgan, our speaker for today's luncheon. So I'm not going to read everything that is on here, but I'm going to get a few of the more salient and more important things. First of all, Dr. Jayne Morgan is a cardiologist and executive director of the COVID Task Force for the Piedmont Healthcare Corporation, the largest healthcare system in Georgia. She has been named to the Health Equity and COVID Task Force for the governor of the state of Georgia and selected to support the Department of Health in its series called Ask the Experts. She is a CNN medical expert contributor and the owner and creator of the Stairwell Chronicles, a social media series directed toward addressing questions surrounding COVID vaccines in a conversational format. Now, what you need to know is, yes, it's sort of like, okay, well, what does she do? Well, she was the director of the cardiovascular research program at Piedmont Healthcare. She was instrumental in helping them expand their research program such that they have patented 2 devices such that they are a significant, rated #1 nationally in this state, in the country for mitral space, mitral valve, I can't do it all, cardiology stuff. Okay, valve stuff. Has to do with fixing tricuspid valves and mitral valves, okay? That's all you really need to know, all right? The other part of this, and this is one of the things that she's here, she's been a dynamic speaker, she's presented for a number of different organizations here in Atlanta, and some important for our discussion here today, she is a board member of the National Diversity and Inclusion Task Force for the American Heart Association. Without further ado, I'd like you to introduce to all of you Dr. Jayne Morgan.
Thank you very much. I appreciate being here today. I have a microphone. I don't know if this can everyone hear me? Okay, perfect. I apologize for being late. I've been on a tour of Atlanta today. It's a lovely town in the rain. But anyway, we made it today, and I hope everybody is feeling good and in a good mood. And so I'll just begin to talk about, or really kind of where we are in this pandemic. How did we get here? What does this mean? What does the future look like? And hopefully answer some questions, a lot of questions that people have. I try to go over as well in my talks. So one of the questions is, how was COVID-19 developed so quickly? And so when we look at the development of COVID-19, COVID-19 is a disease that is caused by a virus called the SARS-CoV-2 virus, severe acute respiratory syndrome coronavirus 2. It is so named, that number 2 at the end is an indicator to the scientific community that this is the second time that we have seen this. When was the first time that we saw it? We initially saw it in 2003. We had a SARS outbreak, severe acute respiratory syndrome. At that time, our global pandemic surveillance teams were in place. We were able to keep that outbreak outside of American shores. But scientific community had alarm bells going off all throughout the community. Why? 1, this is a virus. Viruses are known to replicate and mutate quickly. 2, it now had been seen in humans, this coronavirus family. And number 3 was spread by respirations, by aerosol. We are all Homo sapiens. We live on a single planet, Earth, in the galaxy. Our atmosphere is hydrogen, nitrogen, oxygen. We are dependent on air. So unlike other outbreaks or pandemics or plagues or contagions that might be foodborne or waterborne or fecal-borne or body fluids or human contact or bodily contact, where with some discomfort, we can protect ourselves and we can make choices of how to avoid it. As Homo sapiens, as human beings, we cannot choose to opt out of air. It's not a choice. So alarm bells in 2003 were certainly ringing in the scientific community. We've got a problem. In 2012, we had an outbreak of a sister family in the coronavirus. You may or may not remember, depends on how much you follow science and what you're thinking, but it's called the MERS virus, Middle Eastern Respiratory Syndrome. Once again, our global pandemic surveillance teams were in place. We were able to contain this outbreak to Saudi Arabia. But now we've had 2 shots fired, and the scientific community is in a frenzy. By the time we get to 2020, when we have our very first case of COVID-19 here in the United States, 17 years of rigorous research had been done on this family of coronaviruses, specifically on the SARS family. And when I say rigorous research, all of the preclinical research had been completed. All of the benchwork research had been completed. The entire genomic sequence of this virus, all 30,000 base pairs, had been identified and analyzed. And the mechanism of action of transferring infection to our bodies had already been identified. That spike protein that binds to the angiotensin-converting enzyme receptors of our cells was already known. All of that was completed. We hadn't even had our first case yet. That's because 2 shots were fired in 2003 and 2012, and 17 years we worked to prepare for what surely was coming. So when people say these are miracle drugs, and look at the miracle that has come, this is not a miracle. This is 2 decades of people's lives of scientific rigor and discipline and preparation, and a global effort for what we saw might be the future of human beings back in 2003. So what else happened to make it appear that things were developing so quickly? Well, we get to the vaccine companies, and by and large, vaccines are developed. By and large, vaccines are developed in 3 phases. This is very high level, just general terms, just in general. Phase 1, phase 2, phase 3. In phase 1, we call that the safety phase. We're enrolling patients into trials. We collect all the data. We have a database lock. We analyze all the data. And if the data looks positive, if it looks good, if it looks as if it's going to actually meet our indication, then we get the green light to move on to phase 2. And then we start the whole thing over again in phase 2, but with a larger amount of people. This is our dose finding. We're looking for the right dose. We close out the trial, we examine all the data, and then we get the green light to go to phase 3. The reason that this is such a laborious, meticulous, methodical, and long process is because it costs almost $1 billion to bring a new vaccine or drug to the market. And so CEOs and executives of companies want to make certain, as we go from each phase, that the data looks positive before they invest another few $100 million in the next phase. We call that killing it early. We want to know early that a compound is going to be ineffective so we can kill it off before we've invested more money in it. So every single phase will be analyzed very rigorously before you get the green light to go to the next phase. Well, what happened when our pandemic started? We stacked the phases. All of the phases began to run concurrently. Why did we do that? Because the vaccine companies now found themselves with unlimited resources. And by resources, I mean money. The US government gave the vaccine companies unlimited amounts of money. When you have unlimited money, you can do a lot of things. For one thing, you now can hire more scientists. You can hold other projects and pull key people from other projects. You can open more study sites. You can enroll people more quickly. But most importantly, if it's somebody else's money, it de-risks the process for you. Remember, the reason the process is so methodical is that no one wants to waste a few $100 million and then at the end your compound is worthless. So by stacking the phases, They didn't look at all the data until the very end, when that $1 billion had been spent. And when they looked at all the data was positive, and that's how we got our vaccines. But if the data had been negative, then it's not an injury to the company. It wasn't their money. So families weren't at risk, their employees weren't at risk, the company wasn't at risk of closing. So by the United States government providing unlimited resources, the development process of the vaccine was de-risked. The companies literally had no risk, no skin in the game. They could completely develop their vaccines and get to the end. So that greatly accelerates the process. And then the third thing is, don't forget, we're under emergency use authorization, except now for Pfizer. I'll talk about that. What is the difference between emergency use authorization and FDA approval? Both require all of the safety data, And in science, all and never are two words that are rarely used. If you ever take an exam and they've got all or never, in one of the answers, that's not the right one. But in this case, I'm saying all the safety data. FDA and EUA require the same safety data. What else did they have? All of the efficacy data, meaning how well do these vaccines work? All of that efficacy data had to be presented for both emergency use authorization and FDA approval. What was different? What was different was the third phase, this durability data, this immunity data. For emergency use authorization, you only are required to present 2 months of that data. We only have to know that the compound that you have developed works for 2 months. FDA approval requires a minimum of 6 months and generally up to 1 year. How do we meet emergency use authorization to even be considered? One, human life has to be at risk. It has to be assessed that human life is at risk. And number two, we have no other viable alternatives, medications, nor interventions available to us. So one, human life is at risk. Two, we don't have any options. Then you can go down the emergency use authorization pathway. So at the time that our vaccines became available under emergency use authorization, we knew they were safe, we knew they were efficacious, and we knew they lasted for 2 months. And beyond that, we didn't know. And it was given 2 thumbs up, let's go, and let's try to get as many people vaccinated as we can. We know that it lasts for 2 months, and we continue to collect information. Each month, you have another month of that durability. Oh, it looks like it lasts 3 months. Oh, now we're at 4 months. That's literally why the information was coming in real time. So that is the difference, and this is how these vaccines seemingly came to us so quickly. But it was not very fast. A lot of work was going on. There was a lot of ingenuity. And at the end, there was a lot of resources. And so we have these vaccines. So what do we know about pregnancy and fertility? I get asked this a lot. So I just, you guys aren't asking me. So I'm just giving you the information because I'm asked this question a lot. So when we look at the Moderna trial enrolled 30,000 participants. When we look at the Pfizer trial, they enrolled about 40,000. Johnson & Johnson enrolled about 42,000 people. That's a lot of people. 110,000 people enrolled in these big trials. These are huge trials globally. But pregnant women were excluded from the trials. If you were pregnant, you could not participate in these trials. Not only that, you had to also submit to a pregnancy test. So we had objective information that you were not pregnant. And women had to sign an attestation stating they also would not become pregnant during the course of this trial. So this is the United States of America. What do you think happened? All right? 23 women in the Moderna trial ended up pregnant. 13 women in the Pfizer trial ended up pregnant. 8 women in the Johnson & Johnson trial ended up pregnant, despite all efforts to the contrary. So we call these unintended pregnancies. Not that they were unintended by the women, but that the companies didn't intend for them to happen. But what that gave us was a little cohort of 44 women now who are pregnant, who've all been immunized. And we can follow these people and follow these pregnancies. All of the pregnancies progressed normally. The women were healthy. The pregnancies were uncomplicated. All of the babies have been born and have been normal and thriving, except for one person who had a spontaneous abortion, a miscarriage. When we unblinded that patient, this is a science term, this blinding, it literally means that no one knows what group they're in. So in general, you might have two groups in a trial, one that's getting the real vaccine and one that's getting a dummy vaccine. And nobody knows which it is. The patient doesn't know, and if it's double-blinded, even the doctor doesn't know. The vial will have a code on it, but will have no indication of what it is, and the liquids look exactly the same. So that's called blinding. So when we unblinded the patient, like at the Oscars, you rip open the envelope, and that's the first time you know who the winner is. When we unblinded the patient and broke the code, it turned out that the patient who had had the spontaneous abortion had been in the placebo group, did not receive the actual vaccine. So what else do we know about pregnancy and fertility? When you register when you go to get your vaccine, you register with a V-Safe app. V as in Victor, V-Safe app. You're asked to download this app. In this app, you can report every single side effect that you want to, that you think is related to the vaccine. It's not what your spouse thinks, what your children think, what your neighbor thinks, what your cousin Eddie thinks, what your doctor thinks. It's what you think. If you think it's a side effect, you get to put it in there. You only have to consult with yourself. To date, at the end of August, and I should have checked the numbers today, last day of August, 150,000 pregnant women had registered with this app. Don't forget, pregnant women were excluded from the trials, these huge trials. The trials themselves had 110,000 people in them that excluded pregnant women. Now pregnant women alone are larger than all three trials together. When we looked at the first 30,000 of these pregnant women and looked at things like intrauterine growth retardation, pregnancy-induced hypertension, preeclampsia, eclampsia, spontaneous abortion, it turned out that these 30,000 women who had received the vaccine had a lower incidence of these negative impacts to the fetus than pregnant women in the general population who had not been vaccinated. That led the American College of Obstetrics and Gynecology, the American Society of Maternal Fetal Medicine, American Society of Reproductive Medicine, and the American Academy of Pediatrics to all come out to say, not only do we recommend these vaccines for pregnant women, we also recommend these vaccines for women desiring to be pregnant. The American College of Obstetrics and Gynecology went further to publish outcomes of pregnant women who are infected with COVID-19 and what happens to their fetus, their baby, and the hospital complications that these mothers endure, to demonstrate the risk-benefit analysis just doesn't bear out. What else do we know about pregnancy and fertility? The Johnson & Johnson vaccine is on a different platform. So Moderna and Pfizer, messenger RNA vaccines. Johnson & Johnson is a viral vector vaccine. This is not the first rodeo for Johnson & Johnson. They have built other viral vector vaccines. They have inoculated tons of pregnant women around the world. They are the ones who built the Ebola vaccine. They did the Zika vaccine. All of this was on this viral vector platform that is still being used for their COVID vaccine as well. So Johnson & Johnson had real-world experience already with that platform even before these vaccines became available. You know, one thing that I'm going to add before I go on is just about male fertility, because every now and then I get that answer, you know, you always talk about women, you don't say anything about the men. So here's what we know about male fertility. When you get the vaccine, sometimes as a side effect, you can get a fever. We know that fever decreases the sperm count, and that it takes about 2 months for the sperm count to recover. So if you develop a fever after receiving the vaccine, your sperm count probably dropped for 2 months before it recovered. That happens when you get a fever for any reason at all. And don't forget, if you get infected with COVID-19, you could have a raging fever, worse than a mild fever that you would get with the vaccine. The other thing to remember is that COVID-19, the SARS-CoV-2 virus, binds, that angiotensin-converting enzyme receptor binds to every cell in your body, including all of the cells in your testicles, in your testes. The COVID-19 vaccines, neither the viral vector nor the messenger RNA, have any impact on the testicles whatsoever. So I always want to bring that up to say that's some of the information that we know. And later, if you want to talk about DNA and messenger RNA, I haven't included it in this talk. I'm happy to bring that up as well. Okay, let's talk about some chronic medical conditions. So chronic medical conditions were not specifically included in these trials. They were not studied in these trials. What are we studying? We're really studying the vaccine effectiveness and whether or not you have symptomatic infection. That being said, they were not, unlike pregnant women, they also were not excluded. So what do we have? We have thousands of patients in the trial with hypertension. We have thousands of patients with diabetes. We have tons of patients with chronic heart disease. We have patients with lung disease. We've got HIV patients. We have autoimmune disease patients. We have cancers of every type. All were included, right? All comers. So the data that was submitted to the FDA is data that is relevant to people with chronic medical conditions. Obesity, which is considered a chronic medical condition, and this is a reflection, really, of where we are in the United States, but more than 50% of all enrollees in the trials were obese, by definition. So we've got a lot of information about that. I'm a cardiologist. I'm going to take an example about cardiology. If you have a significant cardiac condition and you contract COVID-19, you're 6 times more likely to be admitted to the hospital than someone else who contracts COVID-19 who does not have a significant heart disease. Once you are in the hospital, you are 12 times more likely to die than someone else in the hospital with COVID-19 who does not have significant heart disease. It is important. Chronic medical conditions decrease your reserve, your physical reserve. And if you are not sure if you have a chronic medical condition, if you take any prescription medication on a regular basis for any reason at all, that's you. That's a chronic medical condition. So when we look at herd immunity, what is herd immunity? We keep talking herd immunity, herd immunity. Herd immunity is a term that came to be at the turn of the century, at the turn of the 19th century. It was used in farmers. Farmers used it. Cattle farmers. They had cows, right? That's the herd. And they noted that when plagues came through and killed their cows, the ones that survived not only seemed to have long-lasting immunity, but cows that they subsequently purchased from other people, and also cows that were subsequently born to the herd, also never seemed to get sick. They started to call this herd immunity. So human medicine adapted that term. That's what we use in human medicine when we talk about our vaccines, but the start was really in farmers. Herd immunity works because as long as a small enough slice of the population remains unimmunized, everyone is protected. The reason for that is if most of us are immunized, then infectious diseases cannot find a host in which to set up a disease process. So people who live within the herd benefit from the fact that disease can't penetrate. And we always depended on that here, not only in the United States, but in the world, because we used live viruses in the past. Our vaccines were live viruses in some form, either killed or attenuated, but they all were developed from live viruses. Many people could not take live viruses. Just a few would be people who are immunocompromised, people on cancer, children with leukemia. If you give them a live virus, that could actually give them the disease and die. So we always relied on herd immunity to protect the vulnerable in our society who were unable to be immunized. So that's what we talk about when we talk about herd immunity. Right now, we are probably looking at trying to reach 80 to 85% herd immunity. And what does that look like? So in the past, when we had, measles, mumps, and chickenpox, and, people may or may not remember, measles used to kill thousands of kids. Like, it was not an innocuous thing. Smallpox as well. And so as we move forward, trying to reach herd immunity, we want to really understand what it means to have protection for yourself and not only for society. And so for people who are not immunized or have not been immunized and may not be moving forward with being immunized, something to remember is that decision, two things. One, is not a singular decision, it's a plural decision because it's a singular decision that has a plural impact. So it's not like choosing whether you're going to have chemotherapy or not, where it only impacts you, or whether you're going to have an x-ray or not. This actually has an impact on other people in society. But also remember that by not taking the vaccine, your very health now is dependent on me and people like me to get our vaccines and stay healthy such that you can stay healthy. And that's how herd immunity works. The other question that I get asked a lot about is, can't we reach herd immunity without getting a vaccine? The answer to that is absolutely yes, we can. And that's what the cattle farmers used to do because they had no other options. But don't forget, if we reach herd immunity naturally, meaning we just allow the disease process to go through society and people develop their own antibodies, we have to be willing to tolerate a certain amount of death. Herd immunity only goes to the survivors of the disease. The ones who died don't benefit from herd immunity. So the vaccines allow us to get to herd immunity, and even our most vulnerable also can reach herd immunity and not die. But we absolutely can do herd immunity without vaccinations. It is not considered the most humane approach because we must be able to tolerate a certain amount of death. So Johnson & Johnson, I always want to talk about this. People ask about it, and it's an opportunity for me to talk about side effects. What did we learn from Johnson & Johnson? Johnson & Johnson is really an excellent vaccine. It is a single-dose vaccine. And it's gotten a lot of bad press, I think, because people don't really understand the science. Johnson & Johnson has an efficacy of 72% with a single dose. Moderna and Pfizer had an efficacy, meaning how well that they worked, with a single dose of only 35 to 45%. That's why they were forced to do a 2-dose trial, because their single dose wasn't strong enough. The FDA requires 50%. That's a low bar. And our flu vaccines, by the way, on any given year are only between 42 and 65% efficacious. That's the best we ever get. So 50% is the bar for the FDA. So Johnson & Johnson cleared that bar with their formulation. Moderna and Pfizer did not. So Moderna and Pfizer had to move forward with developing a 2-dose formulation Whereas Johnson & Johnson was able to do a single-dose formulation because not only did it clear the bar, it cleared it in spades. It was at 72%. That's even higher than our flu vaccines. Unfortunately for Johnson & Johnson, what happened was that by Moderna and Pfizer moving to a 2-dose formulation, their efficacy, which surprised even them and surprised everybody else, ended up in the 90%. And people say, oh, the Moderna and Pfizer vaccines are better. The other thing we have to remember is that Johnson & Johnson was third to the market, and when they trialed their vaccine, it was against a more strident environment of mutations. You may not remember the UK variant was circulating, the Alpha variant. We had the Brazilian variant. We had the South African variant. Moderna and Pfizer didn't have all of that, sir. Those mutations, those variants didn't exist when those trials are being run. So Johnson & Johnson trialed their formulation in a more hostile environment. Moderna and Pfizer, having gone first early in the pandemic, did not have to trial their formulations against these types of variants. And so the thing to remember when you are thinking about these vaccines and looking at all these numbers, this is not math, this is science. The numbers are not indicative of math. So we have to always have the context. The other thing that I will talk about with Johnson & Johnson, we paused the trial because we were having these CVST, cerebral venous sinus thromboses, these special types of autoimmune blood clots that were occurring in the brain. What does that mean? So they stopped the distribution, really to make certain that we weren't missing something and uncovering something big. That's number one. Number two, we wanted to make certain that hospitals and doctors had an opportunity to learn exactly what it is, how to recognize it immediately, and treat it immediately, because it's entirely treatable if you understand what it is. This is not our bread and butter, shall I say, blood clot. So we needed to do autoimmune testing if someone comes in with a blood clot and has had a vaccine, which may not be something that we would think to do. So those are the two reasons that the trial was stopped. It was incredibly baffling to me and other scientists of why you would stop the distribution. We had, I think, a total eventually of maybe 12 or 14 patients out of 7 million distribution. It was just incredible. I've never seen anything stopped with such a low incidence. But that is an indicator of just how many eyes were on it. And they stopped then. And if you don't believe me, birth control pills that we accept and take and prescribe every single day have a risk of a blood clot of 1 in 1,000. Johnson & Johnson is 7 in 1 million. But we stopped that trial, but we kept taking our birth control pills. If you think about it in another way, car accidents, if you're in a car accident, your chance of dying are 1 in 77. Another interesting data point, your chances of getting admitted to the emergency room with a pogo stick injury are greater than getting a blood clot with Johnson & Johnson vaccine. So how did we get here? How did we get here? I've talked about native immunity and vaccine immunity a bit, but let me talk a little bit more about it. So why are we recommending these vaccines even for people who've already been infected? So it's what we call native immunity. You have natural immunity because you actually got infected. So one thing that we have noted with people who have native immunity, meaning they actually were infected with the SARS-CoV-2 virus and have recovered, is that they have great antibodies for 30 days. After 30 days, it drops slightly to about 80 or 81% coverage. After that, there is a separation of the lines. Some people will continue on at this 81% coverage, which is pretty good. Flat line. Others will continue to drop precipitously. So the lines separate. We can't tell who's who. Who's going to go on to longer protection? Who's going to drop off? The other thing is that even those who go on to longer protection at that 81%, our vaccines are covering you at 90%. We want everybody to be as high as possible. So these are the recommendations and why we are seeing them. It doesn't mean they're not going to change. That's one thing about the beauty of science. It evolves. You cannot be in science if you are not a lifelong learner, number one. And so I think this COVID pandemic certainly demonstrates that. So when you look at people with native immunity who've been infected, we are recommending that you get your vaccination within 30 days of your infection. Before we hit that mark where everybody's going to drop, and then some are really going to drop, and others are going to level off. We want you to get your vaccine within 30 days. If you received monoclonal antibody infusions, it's getting a lot of hype in the news, Bamlanivimab, Regen-Co. If you received that Bam, whatever you're calling it, if you receive monoclonal antibody infusion, you'll know that because you got an IV in your arm, and they ran a bag of fluid, so you would probably remember it, and it says an outpatient. If you receive that, then you must wait 90 days before you get the vaccine, because we now have just infused you with a lot of artificial antibodies that won't last long, but if we then have your body stimulated to create its own antibodies, we could run the risk of what's called a cytokine storm. So again, if you've been infected, we want you to get the vaccine within 30 days. If you've been infected and got monoclonal antibody therapy, we want you to wait 90 days. So let's talk about the Israeli study. That's gotten a lot of hype in the news, so I just thought I would, talk about it. I don't know if people have heard about it or not, but Israeli study came out and showed that patients in their society, people in their society who had been immunized were 27 times more likely to be admitted to the hospital with a COVID infection than those who were not immunized. And the conclusion was native immunity is superior to vaccine immunity or some type of sensational headline. So a couple of things when we talk about misinformation. This is not necessarily misinformation, but this is what we call an observational study. And they say that right there. This is an observational study. An observational study in science literally means we have no data. That's what an observational study means. This is not a randomized clinical controlled trial with two groups that we are comparing. This is something that is being observed but not validated. It's not able to be reproduced. For data to be valid, it's got to be able to be reproduced. If they do it in Israel, I should be able to conduct the same trial here in Georgia. They should be able to do the same trial in England, the same trial in China, and we all get the same results. That's reproducible data that we generally comes out of clinical trials. So this was an observational study. The other thing is they use the word study, not trial. So another indicator of where we are going with this. And so many scientists probably wouldn't even read this because just the fact that it's observational, really you can just come to any conclusion, really. It may be true, it may not be. We just haven't shown it to be true. We're not saying it's not true, we're just saying you haven't shown it to be true in an observational study. The other thing to think about, and I've already talked about this with herd immunity, is that they compared two groups. They compared one group that had had a native infection, they had been infected with COVID and never been vaccinated, and the other group that had been vaccinated. What they didn't account for is what I talked with you all about earlier, is that in the native group, they really were only looking at the survivors. They didn't account for everybody who had died from the infection. They only talked about the survivors. They didn't call them the survivors, but you know they're the survivors if they're counting them because they're alive to be counted. So this was a survivor group that they're now comparing to the vaccine group. And the other thing to think about when you look at that study, which is why you really have to make certain you know what you're looking at, they only looked at people in the January and February timeframe of 2020. And don't forget, that's when we were distributing vaccines mostly to people over the ages of 65 and people with chronic medical conditions. So those people were included in the vaccine group because they received protection and were more likely to have breakthrough infections, but were likely to be excluded in the native group because they're the ones who are more likely to have weakened systems and to have died. So once again, it's not math, it's science. We weren't comparing apples to apples, right? These are not equitable groups. This is not a randomized clinical trial. We can just punch holes in everything. But the key takeaway to this is don't forget, that's a survivor cohort. They don't talk about it, but people died to reach that. And then they counted those who were left standing and developed that information. Okay, side effects of the vaccine. More common side effects: nausea, vomiting, chills, fever, headaches, body aches, myalgias, injection site pain. Less common side effects with the messenger RNA vaccines. You've heard about this, maybe lymphadenopathy, which is swelling of the lymph nodes, which we primarily see in women. You can have swelling on the same side of your injection site, under your arm, what we call supraclavicular, on top of your clavicles, your collarbones, or cervical, the back of your neck, on the same side of the injection. Why is this a problem? It is a problem if you have a history of breast cancer. That can be really scary. You start to develop nodes. If you have a family history of breast cancer, or you're in the middle of a breast cancer workup and now you've got swollen lymph nodes. It also can create artifact if you're having any kind of breast imaging and be misread. And then you go on to more and more tests to exclude something that really wasn't real. We call that diagnostic confusion. So we want to make certain that people know, women especially, that you can develop lymphadenopathy. It's not common, but it does occur. And if it occurs in women, it generally resolves on its own within 7 days. We see that only with the messenger RNA vaccines, Moderna and Pfizer. What else do we see with the messenger RNA vaccines? We see this myocarditis, which you may have heard of, inflammation of the lining of the heart. We see that mostly in males, 35 years of age and younger. Sort of like the blood clots, I don't want to minimize anything, it's entirely treatable. So I want to go back to the blood clots for a second because, blood clots are serious for Johnson & Johnson, and myocarditis is also serious. I don't want to minimize that. But I also want to say, if you seek medical attention, it's 100% treatable. So I just want to be clear about that too. So this is the risk that we see: myocarditis, lymphadenopathy, the more severe side effects with the messenger RNA vaccines, and then blood clots that we see with the Johnson & Johnson vaccine. One thing that I forgot to tell you about the blood clots in Johnson & Johnson vaccine, they occur in women between the ages of 18 and 45, and mostly cluster in women between the ages of 34 and 40. That's where most of them happen. And then we had kind of these outliers at the other end of the spectrum. Variance versus mutations. How am I working on my time? I see you stand up back there. Is that a signal? Okay, so just let me know. I can talk forever. I want to just make sure I understand the signals in the room. Variance versus mutations. I want to talk about this for a minute because you may not have recognized that in the news and in print media, we have started to use the word a few months ago, variant. We used to talk about mutations. And we started talking about variants. That maybe was innocuous to you, but it is not inconsequential in science. That denoted an escalation of this pandemic, that one word. Here's the difference. A variant is a cluster or collection of several mutations that have all come together to form a new version of the original virus. That's an escalation from just having mutations. And now we have had several variants. Our most significant one is the one that we're battling now, the Delta variant, a cluster of mutations that have come together to create a new version. This is not the same virus as last year. This is a new version of the original virus. That's what variant means. And the news just says, oh, this variant, oh, that variant, but nobody seems that. That's crazy. So this term is very, very, very significant. So that's the difference when we talk about variants and mutations. And maybe I'll just take a moment here just to talk about people asking me about mu and people asking me about lambda and these other variants that are coming. So we know that the mu variant is here. It's certainly in 49 states here in the United States. It seems to be now at a low level, and it is categorized as a VOI, variant of interest, by the World Health Organization. And by now, you've heard me talk for a few minutes. You know that all of these things mean something, right? They all mean something. So VOI means we're keeping an eye on it. It has characteristics that are troubling, but so far we don't see it taking over. If you ever hear that the mu variant or read that it is a VOC, variant of concern, that is another level up, which means that we now see real movement of this mu variant. The mu variant so far seems to be, as far as contagiousness, infectivity, and severity of disease, somewhere between the alpha variant, which previously, before Delta, was our most strident variant, and the Delta variant. It seems to be falling actually somewhere in between. Not quite as strong as the Delta, more, we call it successful, right? More successful than the alpha variant, not quite as successful as the Delta variant. So that's how we characterize these to look at that. When we look at our fourth surge, which is what we're in now, the fourth surge, this is the first surge that we've had that is entirely preventable. Vaccines were available, they were readily accessible. So this has been the surge that has been the most demoralizing for physicians and hospital systems, because this is the surge that was preventable. When we look at viruses and humans, all forms of life on Earth strive to survive, right? That's what we do. Everything strives to survive. So the viruses want to live, humans want to live. Survival always goes to the fittest. In science, the fittest is not the strongest. The fittest is the most adaptable. The most adaptable species always survives. Viruses are incredibly adaptable. That's why in my first slide back in 2003, scientists got to work. Because when we talk about survival of the fittest, that's what we're talking about. In this fourth surge, that has been entirely preventable. We are seeing, this is when we talk about reproducible information, not just in our hospital system here at Piedmont, but in every single hospital system across the United States. The majority of people who are hospitalized are unvaccinated. It doesn't mean that vaccinated people are not being infected. It just means they're not being hospitalized. So their lives are not at risk. These breakthrough infections that you see a lot of information about, we do have more breakthrough infections with this Delta variant. Why? This Delta variant has a viral load 1,200 times higher than the previous variants. So with the previous variants, you may have remembered that Dr. Walensky, the director of the CDC, said, hey, if you're fully immunized and vaccinated, you can take off your mask. You can socialize indoors and outdoors. The reason for that is that your vaccines were holding and protecting you enough to keep the virus so low in your body that not only would you not get sick, your chances of passing it on to others was also incredibly low. But the game changed with this Delta variant. The viral load was so high that even though you still may not get sick because you have been vaccinated and it's protecting you, you can now pass it on to others. You're carrying so many viral particles that you can pass this on to other people. That's how the game changed with the Delta variant. And that's why we were out of masks, then we went back into masks. So what does that mean for what we are doing as a society, and why do we keep getting these variants? This is entirely behaviorally driven, just like this fourth surge. And we want to reach herd immunity as a society and not community immunity. And I think maybe we can see, if we look at the map of the United States, why community immunity doesn't work. But let me give you an example. I live here in Atlanta. We don't necessarily have a high uptake of vaccines. We're still less than 50%. We have an opportunity for the virus to invade our bodies. Don't forget, human beings are the host. Without humans, the virus dies. It must find us and invade our bodies in order to replicate. And then any errors in replication is what we call mutations. The virus needs us in order to continue to replicate and mutate and learn. It learns as it goes along. Let's say you're living in Vermont or some other state that has a higher vaccine uptake. You've removed your mask, you're kind of moving around pretty freely, and variants aren't really developing there. What happens? Eventually, because we are an interdependent society, people from Vermont mix in with people from Georgia, and everybody gets together, and the mutations that have been allowed to develop in a community with a low immunization rate have the potential to overtake the vaccination status of our vaccines. And then not only are we talking about boosters and third shots, we might be talking about a complete reformulation of our vaccines. And we go back to square one, and round and round and round we go. And so this is why it's been incredibly disheartening this time as we're in the hospitals battling this, people right on the front lines. It's been very difficult, but that's one of the reasons we are striving for herd immunity and not community immunity, because community immunity sets up a nidus or focus of where mutations and variants can develop. Don't forget, I know this seems like 10 years ago, but actually it was just a year and a half ago, when the pandemic started, we were all going to shelter in place for 60 days. The reason for that is such that the virus could pass over and not find any human hosts and then die. It literally cannot live without us. We allow the virus to live. But, you know, we couldn't do it. We were out and about and blah, blah, blah. And here we are a year and a half later with a Delta variant that we didn't, you know, we never even heard of a year and a half ago. So that's where we are with all of these things that are going on. I'm going to end here. I do something called the stairwell chronicles, where I take one COVID question or one COVID vaccine question and I answer it in 60 seconds or less. I talk about it. You see me talking. I call it stairwell chronicles because I literally sit on the steps. I don't have on a white coat. And I just talk to you like your sister, your friend, your neighbor. I demystify science. I just talk about and answer in plain language. It's called the Stairwell Chronicles. You can find it on Twitter, Instagram, or I have a YouTube channel. Feel free to subscribe. On YouTube as well, I have a lot of my media talks as I go on to different medias, local Atlanta and also national and international. And then LinkedIn is Jayne Morgan, MD. So check out Stairwell Chronicles. Look through the whole catalog. Look at the questions. Maybe there's a question that speaks to you. Click on it. It only runs for 60 seconds. That includes the greeting and salutation. So it's only a minute of your time. So if you don't like what I said, you can't be mad. It was a minute. And go on to the next one. And then the last thing I would say, here's an example of some of the stairwell chronicles. The one on the left, I never take vaccines. I'm always healthy. I talk about that. Boosters and the immunocompromised. Now that I'm fully vaccinated, can I transmit COVID to others? So I just answer questions, and I really create stairwell chronicles based on questions that I just hear out in the community. I think, okay, I've heard a couple of people ask me that. Let me create a talking point on it. Let me reduce it to 60 seconds. And if you think that's easy, it's not. You know, I think, what is his name? Tom Sawyer. I'm blanking on who wrote Tom Sawyer. But Tom Sawyer. Tell me who? Thank you, Mark Twain. Thank you. I was blanking on. Mark Twain once said he had written a very long letter to an organization. And then when he got there, at the end of the letter, he apologized and said, I would have written a shorter letter, but I didn't have time. And if you think it's easy to explain something scientific in 60 seconds, it's not, but I make a good faith effort. I've done probably 60 or 70 of them. Feel free to look through my catalogs. I have all kinds of questions. Feel free to reach out to me, you know, social media and say, hey, what about this? And I might do a stairwell chronicle on it. So thank you very much.
Thank you so much, Dr. Morgan. Fantastic talk.
Thank you.
Can we ask one question? Where's Dee? Can she have one question?
It's not my stairwell chronicle, so I can't do 60 seconds, but yes, I might do it, yes.
Ms. Werner.
What? Okay. Yeah, so that's a good question. So she's asking, you know, if herd immunity is 80%, what happens if we get another variant? So remember, at the beginning of this pandemic, we were only trying to reach 70%. Now our stakes are higher. We have to reach 80% because as we get more variants and and they become more infectious, then our herd immunity criteria creeps up as well. So we don't want it to keep going up. We've gone from 70 to 80, but it will continue to go up. It'll be higher and higher and higher what we will need to achieve as we have more and more challenging, shall we say, variants to manage. Thank you.